Hello and welcome everyone to another MCAT ACE lecture series. In today's lecture, I'm going to go over the circulatory system. So this section will be divided into two parts. Part 1 is covering all the topics that are shown in the red box. And then in the second part, which is the second video, we will go over the rest of the topics that are shown here at the bottom. So this first video is going to cover the functions and role of the circulatory system. So the general functions as well as its specific function in thermoregulation. Then we will go over structure of the heart, how it has 4 chambers and how it helps with the circulation of the blood into the systemic as well as the pulmonary circulation. We will go over the difference between systolic and diastolic pressures. As we discussed, we'll go over pulmonary and systemic circulation. Then we will discuss the structure of the endothelial cells, arterial and venous system, as well as capillary beds. And then finally, we will conclude our video by going over the composition of the blood as well as its mechanism of carrying oxygen and carbon dioxide. All right, so with that in mind, let's start the lecture. So going over the functions of the circulatory system. So basically the circulatory system is divided into two parts. Number 1 is the cardiovascular system and number 2 is the lymphatic system. So this first lecture is covering the cardiovascular system. In the second lecture is where we will go over the lymphatic system. So regarding the functions of the cardiovascular system is composed of the heart, blood and blood vessels, and its functions include number 1, it's for the distribution of water, electrolytes, as well as oxygen and other nutrients to different tissues in the body. And then while it's distributing oxygen and nutrients to different tissues in the body, its other function is to take away the waste products, like for instance carbon dioxide or urea. These are being taken away from the tissues to the lungs in order to expel CO2, as well as into the excretory organs, like for example kidneys, for excreting the urea. The other functions of the cardiovascular system is distribution of hormones throughout the body. So all the hormones that are produced in the endocrine glands are being released into the blood, and from there on it distributes to different tissues in the body. The other function of the cardiovascular system is housing the immune cells, which are white blood cells, as well as antibodies to help fight different infections. And then finally, another important function of the cardiovascular system is thermoregulation, or maintenance of the core body temperature. So as you know, the human body requires to maintain a narrow core temperature that is about 36.5 to 37.5 degree of centigrade. And can you tell me why this range needs to be maintained in the human body? So basically, this temperature range is required for the optimal function of the enzymes. And so body has its own homeostasis mechanisms in order to maintain the correct temperature upon changes in the external environment. Like for instance, if the temperature outside changes, the body still needs to maintain this 36.5 to 37.5 degree temperature. And so this function can be done through various different mechanisms. And so the body can lower the temperature, for instance, by sweating, as a consequence of which there will be increased heat loss. And so body temperature will drop. Another mechanism is by shivering of the muscles, as a consequence of which heat will be generated. And then the body temperature increases. And the third important mechanism that relates to the cardiovascular system is by controlling blood flow to the skin. And so if the outside temperature is too low, the cardiovascular system will cause vasoconstriction of the skin vessels in order to let less blood get to the surface, and so this way it will maintain the core temperature. On the other hand, if the body is too hot, then the vessels that are allowing blood to circulate close to the surface of the body or the skin are being vasodilated in order to lower the body temperature. And one mechanism through which the circulation of the blood into the capillary bed is controlled is with the help of the precapillary sphincters. And so these precapillary sphincters, these are actually one-way valves that will control blood flow through the capillaries. And so when we need high tissue perfusion, like for instance, if the muscle is exercising, now we need a lot of blood to perfuse that tissue. In that situation, the precapillary sphincters open to allow perfusion of the blood into the capillary bed. On the other hand, when we want low tissue perfusion, like for instance, with cold temperature, we want low perfusion of the skin. Under those circumstances, the precapillary sphincters close in order not to let any blood to go into the capillary bed. And so this way, the blood will be shunted from arterial directly into venules in order to preserve heat. All right, so that covers the functions of the cardiovascular system. Now let's go over composition of the blood. So if you get 10 ml sample of a blood and you centrifuge that, what you will get is that you will get about 55% of a yellow clear liquid that is called plasma. So this plasma composes 55% of the total blood. And this plasma contains electrolytes, coagulation proteins, albumin, hormones, lipoproteins, and other molecules. So this whole thing is found within the plasma that composes 55% of the total blood volume. Red blood cells, on the other hand, composes about 45% of the total blood volume, and these are composed of red blood cells, the other name for which is erythrocytes. And then there's a small layer in between the plasma and the red blood cells, and that layer contains the white blood cells, which are our immune cells. The other name for white blood cells is leukocytes. And then this layer also contains platelets, which are required for coagulation. Now, the lifespan of these red blood cells is about 100 to 120 days. After which, these red blood cells, the ones that are damaged or the ones that are too old, will be destroyed by spleen. And so spleen is used for erythrosis or destruction of red blood cells. Therefore, the body needs to continuously make new blood cells, and formation of new mature red blood cells is called hematopoiesis. So here we have hematopoiesis, which forms new blood cells. And it's the function of the hematopoietic stem cells, which are a form of multipotent stem cells, to give rise to all these different red blood cells. So here I'm not going to go over the function of every one of these blood cells. In the immunology section, I will go over the function, specific function of every one of these cells. But here, let's focus on the hematopoiesis aspect of blood cell generation. So the multipotent, what's the definition of multipotent? Again, we have different type of stem cells, and in the embryology section of our videos, we have gone over different forms of stem cells. Again, we have totipotent, stem cells, which can form any type of cells as well as extra embryonic structures like for instance placental cells. Then we have pluripotent cells, which can give rise almost to any cells except extra embryonic or placental cells. And then finally we have multipotent cells, which are more limited in the type of cells that they can give rise to. So here we have multipotent stem cells that can give rise to different blood cells, but they're limited. Like for instance, they can't give rise to neurons. Pluripotent or totipotent can do that, but multipotent is limited. So anyway, here we have a case of a multipotent stem cells, which gives rise to different type of progenitor cells. The common lymphoid progenitor cells that give rise to natural killer cell T lymphocytes, which are two type, CD4 positive helper T cells, and then CD8 positive cytotoxic T cells. And the reason here we've shown thymus is that lymphocytes, despite they're being produced in the bone marrow, they need to mature in the thymus. So anyway, we have CD4 positive helper T cells and then CD8 positive cytotoxic T cells. Then we have B lymphocytes, which with the help of the helper T cells, they can convert into plasma cell for antibody production. Again, we will go over the function of every one of these cells in the immunology video of our lecture series. Then we have the common myeloid progenitor cells that give rise to megakaryocytes. And these megakaryocytes are large cells, and they have some blebs that are coming off from their membrane, and these shedding of bleb and cytoplasmic content is what gives rise to platelets. Then we have erythrocytes or red blood cells, and these red blood cells are thinner at the center. So instead of having a spherical shape, they have the biconcave shape in order to allow more surface areas for increased capacity of carrying oxygen. So that's why the center and outside color is different when we are looking at red blood cells. Then we have the mast cells, which release histamine in order to induce an allergic reaction. Then we have the less mature forms of cells that are referred by blast. We have myeloblast, that is the immature form. Once it matures, it becomes granulocytes that gives rise to neutrophils, basophils, and eosinophils. And then we have monoblast that once it matures, it becomes monocyte that gives rise to macrophages, the function of which is phagocytosis of different pathogens. So let's not worry about the function of every one of these cells. I will cover them in a different lecture, but that was basically the breakdown of the hematopoiesis process, how different red blood cells are arising from these hematopoietic stem cells, which are multipotent. All right. Now, production of red blood cells in an adult is primarily by the bone marrow. So here you can see a fetal age. After age 40, it's the bone marrow that takes over and does all the red blood cell production. But prior to that, before the bone marrow actually reaches the level to take over all the red blood cell production, it's the function of the yolk sac at the beginning of fetal life, and then later on liver and spleen will produce some of the fetal blood. And then towards the end of the fetal life, bone marrow will take over. And so early in life, the production of red blood cells in the bone marrow can be done in the long bones, but then later in life, the long bones assume the weight-bearing function, as a consequence of which the bone marrow in the long bones turns yellow. So subsequently, hematopoiesis shifts to red marrow of axial skeleton as we age. And axial skeleton includes, for instance, skull, pelvis, ribs, and other flat bones. I would also like to mention that while it's the function of the bone marrow to produce red blood cells, when there is high demand for production of red blood cells, or when there is, for instance, problem with the bone marrow, that's the time that the spleen and liver can again start production of red blood cells. So while liver and spleen are primarily required for production of red blood cells during the fetal life, later on during adulthood, if there is a problem with the bone marrow, then liver and spleen can start production of red blood cells again. All right, now let me ask you a question. Which of these organs or tissues are responsible for destruction of red blood cells? We just talked about production. How about destruction? So spleen is primarily required for destruction of red blood cells. And then other organs, like for instance liver or the bone marrow, can also destroy red blood cells, but again spleen is the major one. Now let's go over destruction of red blood cells. We just mentioned that red blood cells can have 100 to 120 days of lifespan in the circulation, after which the old red blood cells or damaged ones need to be removed. And so before we go over the mechanism of destruction, I would like to first go over structure of the spleen. So the spleen is made of a red pulp, and its red color is coming from too much blood that is engorging this tissue. Then we have the white pulp, which mainly contains B cells. So a form of white blood cells is in the white pulp. And then finally we have the splenic sinusoids. And these splenic sinusoids are lined with macrophages. All right? So macrophages are lining the splenic sinusoids. And when red blood cells are squeezing through the splenic sinusoids, it's the surrounding macrophages that will destroy any abnormal or damaged or aged red blood cells. And so under these circumstances, macrophages will phagocytose the red blood cells, and the red blood cell degradation products will give rise to heme and globin component. The globin will be further degraded into amino acids, which will be taken up by the rest of the body in order to produce more proteins. The heme, on the other hand, is broken down into iron, which can be stored with different proteins, like for instance ferritin, transferrin, and hemosiderin, and the porphyrin component, which is broken down into unconjugated bilirubin. And this unconjugated bilirubin is water insoluble. And so in order for it to get transported to the liver, unconjugated bilirubin will bind to albumin, and it gets transported to the liver. And inside the liver, bilirubin becomes conjugated. So conjugated bilirubin will be formed in the liver, and this conjugated form is now water soluble. And so this water soluble form can be excreted inside the intestine, and from there on it will be excreted in the feces. Some of this conjugated bilirubin will also be taken up by the blood and be excreted by the urine. Now I would like to remind you that the bile is also playing an important role in emulsification of fats. So some of these bile components will be stored in the gall bladder and will be used as bile salts for emulsification of fats. And again, that topic will be discussed extensively once we are going over the gastrointestinal and the digestive system. All right, and so that covers the destruction of red blood cells by spleen. Next, I would like to go over regulation of the plasma volume. So once the plasma volume needs to be increased or decreased, there are different mechanisms. Afferent or sensory pathways detect decrease or increase in the plasma volume. And then the efferent or motor pathways will induce changes in order to correct for that abnormality. Now I would like to mention here, I'm going over the plasma volume, which can affect the blood pressure. So as we increase plasma volume, blood pressure also increases. But there are more than just regulating the plasma volume for regulation of blood pressure. We can vasoconstrict or increase contractility of the heart in order to increase blood pressure. And those mechanisms will be covered in the second part of this lecture. So here we are only going over the regulation of the plasma volume. And so I'd like to go over the afferent as well as the efferent pathways that helps in the regulation of plasma volume. Okay, so let's first start with the afferent pathways, and these afferent pathways sense changes in the blood volume and there are different mechanisms that they can use. So the first mechanism is the cardiac atria that contains mechanoreceptors. And these mechanoreceptors can detect decrease or increase in the pressure that is being imposed on the cardiac wall, as a consequence of which under circumstances where there is too much pressure, there would be increased production of the atrial natriuretic peptide by the wall of the atria. Inside the heart. And the function of the atrial natriuretic peptide is to increase glomerular filtration rate in order to increase excretion of the body fluids. It also decreases the production of renin in order to reduce activation of the renin-angiotensin-aldosterone system. And then finally, it can induce the vasodilation by activation of the sympathetic nervous system. All right, so this was the first mechanism that increased blood pressure can be compensated for by increased production of the atrial natriuretic peptide. The second mechanism is by mechanoreceptors that are found in the aortic arch as well as the carotid sinus. And these mechanoreceptors again detect changes in the blood pressure and regulate the activities of the sympathetic nervous system and the renin-angiotensin-aldosterone system. So once there is too much pressure inside either the aortic arch or the carotid sinus, there would be decreased activation of the sympathetic nervous system as well as decreased activation of the renin-angiotensin aldosterone system. And then I'll go over these mechanisms in a separate video again. And then there will be also decreased production of vasopressin. All right, so all these hormones are covered in our renal videos as well as in the endocrinology section, going over the function of different hormones. But we will briefly touch on here in this lecture later on as I'm going through the efferent pathways. All right, and then finally, the third mechanism for sensing changes in the plasma volume is by the juxtaglomerular apparatus. So renal juxtaglomerular apparatus, which detects a decrease in blood pressure or a decrease in sodium concentration, as a consequence of which, so once there is a decrease in blood pressure or decrease in sodium, it will induce secretion of renin, as a consequence of which it will activate the angiotensin-aldosterone system. All right, so here we had the first mechanism, cardiac atria, which detects high pressure in atrial wall, as a consequence of which increases the atrial natriuretic peptide in order to decrease plasma volume. While the other two mechanisms that are discussed, they would activate the renin-angiotensin-aldosterone system in order to increase the plasma volume. All right, we are done with the efferent pathways. Now let's move on to the efferent pathways. All right, so the first hormones that we are having here are working as natriuresis, where in response to the stretch of the cardiac wall, there will be increased production of the atrial natriuretic peptide as well as brain natriuretic peptide. And these are secreted from the atrial as well as the ventricular myocardium. So atrial natriuretic peptide is being secreted from the atrium, and brain natriuretic peptide is secreted from ventricles. I know it sounds strange. Why do we say brain? Because initially they thought it's being secreted by brain, but then later on they found out that ventricles is the main source of the brain natriuretic peptide. So once these natriuretic peptides are produced, they would act directly on the renal system in order to increase the glomerular filtration rate, as a consequence of which so much of the blood filtrate will be excreted in the urine. So it will increase urine production, as a consequence of which it will decrease plasma volume. These hormones also decrease activation of the sympathetic nervous system in order to cause vasodilation and decrease blood pressure, and then they would also decrease activation of the renin. Okay, and that would again lead to decreased plasma volume. The next system is the sympathetic nervous system, and this sympathetic nervous system can increase the renal reabsorption of sodium, so sodium reabsorption is increased in the kidneys, and it will also stimulate the renin, angiotensin, aldosterone system, which is discussed right here. All right, now let's go over the renin, angiotensin, aldosterone system. All right, so basically this system uses multiple hormones to regulate blood pressure, volume, as well as osmolarity. The initial product of this system is being produced by the liver, and it's called angiotensin. And then in response to low blood pressure in the juxtaglomerular cells or low sodium in the macula densa of the distal convoluted tubule. So here we have two mechanisms. One is low blood pressure in the juxtaglomerular cells. And then we have low sodium that is detected by the macula densa of the distal convoluted tubules of the kidney. And then in response to these changes, renin will be produced to convert angiotensin into its active form, which is angiotensin 1 and then later on angiotensin 2. Once angiotensin 2 is produced, it will cause vasoconstriction of the blood vessels in order to increase blood pressure. It will enhance production of the aldosterone by the adrenal cortex. So just pay attention, it's an important gland that you should memorize. Aldosterone is produced by the adrenal cortex, which is located right above the kidney. And then the function of the adrenal cortex is to increase the sodium absorption and decrease potassium reabsorption. Right, so increase sodium reabsorption in exchange for decreased potassium. And now since there is increased sodium reabsorption, water will follow sodium, as a consequence of which there will be increased plasma volume and increased blood pressure. And then finally, angiotensin 2 acts on antidiuretic hormone, the other name for which is vasopressin, which are produced by the posterior pituitary gland. And the function of these hormones is to incorporate aquaporin, water channels inside the collecting ducts of the renal tubules in order to increase water reabsorption by the kidneys, as a consequence of which blood volume and pressure increases. So therefore, conditions that will stimulate production of vasopressin or antidiuretic hormone includes, like for instance, dehydration or increased osmolarity of the body extracellular fluids. All right. The next section is moving on to the structure and function of the heart. All right, so the heart is a muscular pump that helps circulate the cardiovascular system, which includes the pulmonary circulation as well as the systemic circulation. And we will go over those in a different image that clarifies the exact difference between pulmonary and systemic circulation. So the heart has 4 chambers. 2 of these chambers are called atria, like for instance the right atrium and the left atrium, and the function of these atria is to pump blood into the ventricles. And then we have 2 ventricles, the right ventricle and the left ventricle. Left ventricle is much more muscular than right ventricle because the function of left ventricle is to pump blood into the systemic circulation. And then the right ventricle, its function is to pump blood into the pulmonary circulation, which is basically the lungs. Now here there are different valves that help prevent backflow of blood into the structures that are pushing the blood through. So here you can see that between the right atrium and the right ventricle, we have a valve here. And can you tell me what's the name of this valve? It's called tricuspid valve. So here we have a tricuspid valve that prevents backflow of blood from right ventricle to the right atrium. And then right ventricle pumps blood into the pulmonary artery. And here again we have a valve called pulmonary valve. Then from here on, blood goes to the lungs. And then from lungs, the blood returns via the pulmonary veins to the left atrium. Left atrium pumps blood into the left ventricle. And here again, we have a valve called bicuspid or mitral valve. And then finally, left ventricle pumps blood into the aorta. And so therefore, we have another valve here that is called aortic valve. And then everything is also listed here. All right, so here, let's discuss what happens when someone injects some drugs into their veins, for instance. This way we can go over the mechanism of how blood circulates the body. So here, let's say we are injecting some drugs into the vein. What happens is that all the blood from the venous system will collect into the vena cava. And vena cava, we have a superior vena cava that collects all the deoxygenated blood from the upper part of the body, and we have the inferior vena cava that collects all the deoxygenated blood from inferior part of the body. And then the blood from vena cava empties into the right atrium. Right atrium pumps blood into the right ventricle, and any backflow of blood is prevented by the tricuspid valve. Now, the function of right ventricle is to pump blood into the pulmonary arteries. So right ventricle pumps blood into the pulmonary arteries. So just notice that even though we have the name artery here, this is deoxygenated blood. So any vessel that leaves the heart has the name artery, but it doesn't necessarily mean that all arteries are carrying oxygen. Here is an example where pulmonary artery is having deoxygenated blood. And so deoxygenated blood will reach the lungs, they will gain oxygen and lose CO2. Now this blood will return to the heart via the pulmonary vein. Again, any vessel that returns blood to the heart is called a vein. But pulmonary vein, since it's coming from the lungs, is rich in O2, or in other words, this is carrying oxygenated blood. So oxygenated blood via the pulmonary vein returns to, can you tell me where? The left atrium. Left atrium pumps blood into the left ventricle, and then bicuspid or mitral valve prevents backflow of blood. Then left ventricle finally pumps blood into the aorta. All right, so left ventricle pumps blood into aorta, and aortic valve prevents backflow of blood, and now blood enters the systemic circulation. All right, so we have the systemic circulation where via the left ventricle the blood is pumped into every tissues in the body except for the lungs. And then we have pulmonary circulation where via the right ventricle blood is pumped into the lungs. All right, and I will go over that again in a separate image that better shows the difference between systemic and pulmonary circulation. All right, now let's go over the mechanism of heart contraction. We were just discussing how heart is pumping the blood, but how is it actually being contracted? And so cardiac muscle is an example of a striated muscle that contains pacemakers. And then, can you tell me what's the definition of pacemakers? So pacemakers are cells, are specialized tissues that produce action potential by themselves. So if you cut all connections to the heart, since heart contains its own pacemakers, it will continue to beat. And so it's because of the function of the pacemakers. So therefore, cardiac contractions are self-driven. But then there are also autonomic connections to these pacemakers. And these are called sympathetic nervous system. So sympathetic nervous system will increase the heart rate. And so here we have increased heart rate. On the other hand, we have the parasympathetic nervous system, which will decrease the heart rate. And the nerve that you should definitely know as a parasympathetic nervous system that decreases the heart rate is called vagus. All right, so these autonomic nerves are affecting the function of the pacemakers in order to regulate rate of heartbeat. All right, now with that information in mind, let's go over let me erase all this. All right, very good. Now let's go over the mechanism of heart contraction. So first we have the sinoatrial node. So here I wrote as SA because it's a sinoatrial node. And it contains pacemakers that initiates the depolarization impulse for the cardiac contraction. And so once there is depolarization, the impulse will travel through the atria until they reach the AV node, the other name for which is atrioventricular. And these are also pacemaker cells. So because AV node is located between the atria and the ventricle, the name atrioventricular or AV node is given to it. All right. Now, what happens if this SA node is destroyed? Well, SA node has a higher rate of firing than AV node. But if it's destroyed, AV node is still a pacemaker cell, so the heart will continue to beat, but its rate, its default rate, will be decreased because AV node doesn't fire as fast as SA node. Of course, still SNS system can act on it to increase the heart rate, but my point is that SA node, since it has faster firing rate, it's primarily determining the rate of the heart rate, the basal rate of the heart rate. And then other factors like SNS and PNS can also affect heart rate. All right, now once the impulse reaches the AV node, there will be a delay before the signal can be further transmitted through. And this is actually great because now there is depolarization of the atria. The atria are contracting, but time is needed until all the blood enters the ventricle. So this delay in the AV node allows enough time for the blood from atria to enter the ventricles. And then from the AV node, there will be impulse passing through the bundle of His, which is divided into the left and right bundle of His. And then bundle of His, with the help of the Purkinje fibers, they will transmit all the impulse through the entire ventricles. So here you can see that the bundle of His, let me come a little lower. So here you can see that the bundle of His allows the action potential to spread to the heart apex, and from here on, with the help of the Purkinje fibers, action potential spreads to the rest of the ventricular muscles. Now the cardiac muscles are linked to each other by intercalated cells which have many gap junctions, as a consequence of which there is a rapid spread of action potential. So gap junctions play an important role in both smooth muscles and cardiac muscles for rapid spread of the action potential. They're not present in skeletal muscles, but for cardiac and smooth muscles, they have a very important role. All right, now before I move on to mechanism of pacemaker activation, I'd like to review one more concept here with you, and that is an EKG. So I'm sure that all of you have seen EKGs, but what do each of these signals mean? And so you know how we have first atrial depolarization that spreads into the ventricles and cause ventricle depolarization. So this is basically what is showing us here. P wave is representing the atrial depolarization, and then QRS is representing the ventricular depolarization. Now, after there is a depolarization, there should be repolarization in order for the membranes to reach their original resting potential. So here, the atrial will undergo repolarization, but the repolarization phase is falling somewhere under QRS, so we won't be able to see the atrial repolarization. The ventricular repolarization, on the other hand, is shown by the T wave. So T wave is representing ventricular repolarization. All right, so let's go over all these one more time. We have the P wave that is representing atrial depolarization. We have QRS that is representing ventricular depolarization. And then finally we have the T wave that is representing ventricular repolarization. Atrial repolarization, we can't see it in this graph because it falls under the QRS interval. All right. Now, in the neurology videos, we discuss how the neurons undergo firing. So first we have a depolarization phase where the sodium will increase the resting membrane potential. And then finally we have the repolarization phase where the potassium channels open in order to decrease membrane potential. Cardiac muscles are being contracted through a very similar mechanism, except they also depend on calcium channels for the repolarization phase. Pacemakers, on the other hand, are kind of different. So for their depolarization phase, they depend on calcium, and for repolarization phase, they depend on potassium channels. Now, here is the funny part, that they have a sodium channel called IFNA, and this F stands for funny. And the reason for that is that these sodium channels keep on leaking. And so that's the reason that the pacemakers keep firing. The reason for that is since these IFNA keep leaking, they will reach a threshold that it induces an action potential. And then there is repolarization, but then the membrane potential increases and there's firing again. And then increases resting membrane potential until there's firing again. Now this pacemaker activity, therefore its rate of firing, is dependent on the IFNA channel. Now, how does the SNS and PNS systems work? So the sympathetic nervous system will increase the slope of the IFNA. So once the slope increases, there will be faster firing. All right, and so that's how sympathetic nervous system increases the heart rate. The parasympathetic nervous system, or the vagus nerve, on the other hand, decreases the slope of IFNA. And so it will take longer time before the threshold is met. It takes longer time before a threshold is met. And so therefore, parasympathetic nervous system, or the vagus nerve, decreases heart rate by decreasing the slope of the IFNA. All right, and so that covers the mechanism of cardiac contraction. Let's move on to the difference between systolic and diastolic pressures. All right, so once the heart contracts and the left ventricle pumps blood into the aorta, the pressure rises from 0 to 120 millimeter mercury. And this is called a systole or systolic pressure. And then the function of the systolic pressure is to pump blood into the systemic circulation. Now, after that, once the aortic valve closes and the heart relaxes, there is a period that is called diastole. And during this period, it's the recoil property of the aorta that maintains the pressure. And so during this period, the diastolic pressure is maintained. And it's usually in healthy people, it's around 80 millimeter mercury. And so when you hear that someone says my blood pressure was 125 over 82 millimeter mercury, for instance, they're talking about systolic blood pressure here, and then the diastolic blood pressure after that. And then the difference between these two is called pulse pressure. Now let's talk about how we can measure the systolic and diastolic blood pressure. So once you place a cuff pressure on the arm and you inflate it, what happens is that the pressure will eventually raise above systolic blood pressure, as a consequence of which the artery will be so squeezed that no blood can pass through. So once you're listening with the stethoscope, you don't hear any sound. So here, let's say that we have the pressure here and you don't hear any sound. Now you will start to slowly release air from the cuff, and then the pressure here, you can see that keeps dropping on and on and on until you reach a point that, like for instance right here, that you would start to hear the heart sound, because now the blood can pass through. So this very first heart sound, which is shown here about 110, represents your systolic blood pressure. And then you would continue doing the same thing. You will continue to let the pressure drop until you reach a point that you don't hear the sound anymore. So the heartbeat sound will disappear. And this second point where the sound disappears represents the diastolic pressure, where in this situation it's about 70. So this patient has a blood pressure of about, let's say, 110 millimeter mercury systolic over 70 millimeter mercury diastolic pressure. All right. The next section is going over the structure of different blood vessels. So we start with arteries, like for instance aorta, that divides into arterioles, then they divide into capillaries, and here we just discussed how the precapillary sphincters can control the amount of blood that is perfusing the capillary bed. And then the capillary blood will empty into venules, and then the veins, which will return into the heart. Now let's go over the actual structure of every one of these vessels. So the vein and the arteries, both of them have three different layers, starting with the tunica interna, which is made of the endothelial cells. And these endothelial cells, they line the lumen of the vein, artery, as well as the capillary. So endothelial cells is found in all three types of vessels. And the function of the endothelial cells is, number one, nutrient and gas exchange. They're also important for formation of new blood vessels, and then they also activate the coagulation or clotting mechanisms. The second layer is tunica media, which contains the smooth muscles, and these smooth muscles are under the control of the autonomic nervous system, like for instance the sympathetic or parasympathetic nervous system, as a consequence of which the SNS system can act on the smooth muscles via the norepinephrine neurotransmitter that is released by SNS and cause contraction of the smooth muscles, as a consequence of which the diameter of the vascular lumen will decrease, and so therefore blood pressure will increase by activation of the sympathetic nervous system. And then the final layer that we have here is called tunica externa, which is made of the elastin and collagens. And the function of tunica externa is basically to protect and reinforce the vascular wall. This layer may also contain nerve fibers or lymphatic vessels, as well as the vasa vasorum. So nerves, lymphatic vessels, as well as vasa vasorum, which is a network of capillaries that is used for nourishing actually tissues with large vessels. And these are all found in the tunica external layer. Now, what I would like you to note here is that the lumen of the veins is much larger than the arteries. And then the structure of the wall, the venous wall, is much thinner and less elastic than the artery, as a consequence of which it allows the veins to stretch and hold a great volume of blood. So you may wonder, where does the 5 liters of blood in our body, where does it go to? Is it stored in the heart? Is it stored in the spleen? So the answer is most of it is being stored in the veins because they have a lot of capacity to stretch and hold a great volume of blood. And then here I have shown you that vena cava, for instance, it's a little less than 3 centimeter versus aorta that is less than 18 millimeter diameter. All right, now let's go over the function of every one of these vessels. So arteries, these are made of a thick elastic wall. So these have a thick and elastic wall that carries blood away from the heart, example of which includes aorta. And this thick and elastic property of these vessels allow them to tolerate the high systolic blood pressure that is pumped with the left ventricle. And the blood from arteries will enter the arterioles. These vessels are very important for regulating blood flow to tissues by constriction or dilation of the vascular lumen via the smooth muscle. So basically the blood pressure is regulated by these vessels. And so the other name that sometimes the arterioles are referred to is called resistance arteries. It's because the resistance of these arteries is what decreases blood flow to different tissues. Then the blood from arterioles will enter the capillaries, and capillaries constitute the smallest division in the blood circulation network. So this is the smallest division. And as we just discussed, the capillaries do not have tunica media or externa. They only have a thin layer of overlapping endothelial cells, and this property is actually important in order to allow easy exchange of gas and fluids and other small molecules between blood and the body cells. Because assume if the capillaries, for instance, had the smooth muscles or the tunica externa, then it wouldn't have been easy for molecules to go across the endothelial cells. But this actually thin layer of overlapping endothelial cells allows easy exchange of gas and other nutrients. Right? Then the blood from capillaries will enter the venules, and these venules are generally the site that white blood cells will exit blood and fight infections. And then finally, venules will empty into veins, which are made of a thin walls, and as we just discussed, they are highly distensible and can hold large blood volume. Actually, as much as 70% of the blood in the circulatory system can be found in our veins. All right, now the veins carry the blood back to the heart, but then there is one major issue here, and it's that the pressure in the vein is dropping. And so how can the blood be lifted from our legs, for instance, all the way to the heart? Because the smooth muscles in the veins is not as strong as arteries. And the answer is via 3 different mechanisms venous blood can return to the heart. So number 1 is by contraction of the skeletal muscles that surrounds the vein, and so therefore contraction of these muscles will help push the blood inside the veins upward towards the heart. The second mechanism is presence of the one-way valves that will prevent backflow of blood against gravity. So once blood is lifted upward, it can't backflow because of the presence of these valves. And then the third mechanism is our respiratory pump, or in other words, our lungs, which will create a negative pressure in our thorax or in our chest during inspiration, as a consequence of which blood is sucked upward. So once we are breathing and we are expanding our lungs, there's a negative pressure that allows air to go inside the lungs. At the same time, this negative pressure also helps with pushing the blood upward towards the heart. All right, so here is the summary of the difference between pulmonary and systemic circulation. So as I just mentioned earlier, that I was going to show you in a separate image, the difference between these two circulations. So the blood, the venous blood from the systemic circulation enters the right atrium and then the right ventricle, and here via the right ventricle, blood enters the pulmonary circulation. So within the pulmonary circulation, blood becomes oxygenated, it enters the right atrium, and then via the right ventricle, blood is again pumped into the rest of the body. And this is called the systemic circulation. So you can see that the left ventricle pumps blood into the systemic circulation. It contains oxygenated blood, while the right ventricle is pumping blood into the pulmonary circulation, and it contains the deoxygenated blood. Right, and now regarding the nutrient and gas exchange, so capillaries, they're only made of a thin layer of endothelial cells. They allow water, glucose, oxygen, and amino acids to enter the tissues, and then the waste products like carbon dioxide, urea, water can re-enter the capillaries, and via the veins they return back to the heart. Same happens in the lungs, where the oxygenated blood, so our veins that contains the oxygenated blood contains about 40 millimeter mercury of oxygen and 46 millimeter mercury of CO2. Once they exchange gas with our alveoli, the oxygen content raises to 100 millimeter mercury and the venous content drops to 40. And then once it passes through the systemic circulation, again, the O2 drops and CO2 increases. Now here via the pulmonary circulation, again, oxygen is recirculating inside the blood. Now you can see that oxygen transport by the blood is one of its very important functions. And so the question is, how does blood carry oxygen? And so there are two different forms that that oxygen is being transported in the blood. Number one, oxygen can actually get dissolved in plasma. So you know how there is so much oxygen that is dissolved in water, and all the amphibian and like fish under the water can use up that oxygen. Same thing can happen with our blood. Oxygen can actually get dissolved in plasma, and this is actually referred to as partial pressure of oxygen in our blood. Alternatively, oxygen can bind to hemoglobin, and this mechanism, the second mechanism, accounts for 98% of the oxygen that is carried by blood. So this is the main mechanism. So let's go over how hemoglobin is transporting oxygen. All right, so hemoglobin, it's an oxygen transporting metalloprotein. It has three fundamental constituents. It contains iron, it contains porphyrin, and it contains globulin. And we just discussed how these components are being produced once a red blood cell is destroyed by the spleen. Now this porphyrin ring has a central ferrous, which is Fe2+, atom that is referred to as a heme. And this heme can reversibly bind to oxygen. All right, now each hemoglobin is made of two pairs of globulin chains, alpha 2 beta 2. It has 2 alpha components and 2 beta components. So a total of 4 heme are being inside each adult hemoglobin. As a consequence of which, each hemoglobin is capable of binding up to 4 oxygen molecules. Now you might have noticed here I'm showing you the oxygen dissociation curve for hemoglobin. You can see that under low partial pressure of the oxygen, the saturation of hemoglobin is low. But as we are increasing the partial pressure, there's all of a sudden like sudden binding of oxygen to the hemoglobin. And this is due to the cooperativity phenomenon, where hemoglobin affinity for oxygen increases after binding of the first molecule. So once the first molecule binds, all of a sudden the rest of the molecules like to bind to oxygen. And this is due to the fact that binding of the first oxygen molecule to a heme group induces a conformational shift that changes the shape of a hemoglobin from taut to a relaxed state, as a consequence of which subsequent O2 binding could be done easier, and so therefore there's a rapid increase in the oxygen binding to hemoglobin after slight increase in the partial pressure of the oxygen. Now here you notice that once the partial pressure of oxygen is low, hemoglobin likes to release O2. And so basically in any tissue that there is demand for oxygen, oxygen will be released. On the other hand, if we are inside, here let me show you with a different color. If we are inside tissues that have high oxygen contents, like for instance inside the alveoli of the lungs, then the hemoglobin likes to bind to the oxygen and hold on to them until they go back via the systemic circulation into tissues that require oxygen, and under those circumstances they will release oxygen. Now there are factors that can affect the affinity of hemoglobin for oxygen, like for instance elevated CO2 content, increased altitude, or 2,3-diphosphoglycerate, which is a byproduct of the glycolysis. So if there is not enough energy production and we have to rely on glycolysis for energy production, it means our body needs oxygen. So increased byproduct of glycolysis, which is 2,3-diphosphoglycerate, increased metabolic activity, or increased H+, like in our muscles. Once there is increased lactic acid production, then that's the time that the oxygen hemoglobin dissociation curve shifts to the right. And what happens once it shifts to the right is that even for a little higher than normal O2, the hemoglobin is willing to release oxygen. So there are factors that shifts the oxygen hemoglobin dissociation curve to the right in order to increase the release of oxygen. Fetal hemoglobin, on the other hand, it has high affinity for oxygen, and the reason for that is that it has to rub the oxygen from maternal blood. It has to have higher binding in order to take it away from mother's blood. And so therefore, once we are comparing the fetal hemoglobin to our normal hemoglobin, its dissociation curve is shifted to the left because it has higher affinity for oxygen. All right. And then finally, high temperature, like for instance from increased metabolic activity in our muscles, induces shift to the right in order to release oxygen. All right, the last topic for this first section of our video is going over the mechanisms of carbon dioxide transport in the blood. We just discussed how oxygen is being transported. Now let's discuss how carbon dioxide is being transported in the blood. So there is a large amount of carbon dioxide that is generated in our muscles as well as other tissues, and they need to be transported back to the lungs. As we just discussed over there, CO2 will be lost and then oxygen will be picked up. But then how does carbon dioxide how is it being transported by the blood? And there are three different mechanisms. The first one, similar to oxygen as we just discussed, CO2 can get dissolved in blood. And this contributes to about 5% of total blood CO2 content that is being transported by the blood. The second mechanism is carbon dioxide bound as carbonate. So inside the red blood inside the red blood cells, there is an enzyme called carbonic anhydrase. So carbonic anhydrase combines CO2 and water in order to convert them into carbonic acid. So water and oxygen, water and CO2 are being combined with the carbonic anhydrase in order to form carbonic acid, or H2CO3. And then H2CO3 will dissociate to form bicarbonate. So here we have bicarbonate, or HCO3 negative, as well as the hydrogen ion, or H+. Now, what happens next is that HCO3 increases inside the red blood cells. There will be exchange of the chloride with HCO3. So chloride ions shifts into the red blood cells. So chloride ions enter the red blood cells and HCO3 will exit. And this phenomenon is referred to as chloride shift. And then once the red blood cells reach the lungs, the reverse process happens. The opposite process, where bicarbonate will shift into the red blood cells and chloride will exit. Now bicarbonate will combine with the hydrogen ion in order to form the carbonic acid, and then carbonic acid will dissociate into water and CO2, and CO2 will be expelled into the alveoli. And then the final mechanism is CO2 bound as carbamate. So basically carbon dioxide can bind to hemoglobin as carbamate, or NH2COOH. But then this binding depends on whether already that hemoglobin has picked up oxygen or not. If that hemoglobin has not picked up oxygen, then CO2 can bind as carbamate. If it has already picked up, then there will be lower affinity. All right, so therefore, we have three different mechanisms for carrying CO2 by the blood. One is being dissolved in the blood, two, carrying it as carbonate, or three, by binding of CO2 as carbamate to the hemoglobin. All right, so basically that concludes the first section of our discussion of the circulatory system. So this was basically entirely the cardiovascular system that we have discussed. We will continue the cardiovascular system in our next lecture, and then finally conclude our discussion with the lymphatic system and some practice questions. All right, so thank you all for listening, and we will see you for the second part of this video.